SwayamPrabha. Digital India. Educated India.
Welcome to the Proteomics course. In the last few lectures, we have discussed about gel-based proteomic techniques. You have seen how two-dimensional electrophoresis can be performed. There are certain limitations of this technology. So, then there are new creative ways, more advanced methods have emerged to overcome those challenges. In the last class, we talked about difference gel electrophoresis or DIGE, and you have seen how to perform the DIGE experiment in detail. So, the previous lecture we discussed about different challenges which are associated with two-dimensional electrophoresis. I gave you few examples how sample preparation, different type of reagents or even isoelectric focusing settings, all of those factors can contribute towards a very bad gel. So, if you have performed everything properly and all your reagents and your protocols are good, at the end you can expect a good gel. But often there are various gel to gel artifacts and variations in traditional two-dimensional electrophoresis method. So, we discussed about how to overcome those methods, those problems. New methods such as DIGE have emerged. So, in today's lecture, we will continue our discussion on gel-based proteomics, certain advantages and challenges of performing DIGE experiments, how to analyze DIGE data by using few very specialized software, and how to interpret that data, how to obtain some meaningful biological information from those analyses. So, today I have invited a guest, Dr. Srinivas from G Healthcare, who is going to talk to us about DIGE technology and give us a demonstration on software to perform DIGE gel analysis. This is my great pleasure to introduce Dr. Srinivas Rao from G Healthcare. He is an application specialist in the research product division of G Healthcare. Dr. Srinivas, thank you for coming for discussion about 2D and DIGE technologies. So, what are the major advantages and disadvantages you see by using 2D approach?
It's very user friendly as well as very low instrumentation cost. There, the complete proteomic, the differentially expressed proteins we can see exactly during this complete 2D electrophoresis technique, as well as the novel proteins we can able to isolate from from this technique. This we can identify very easily the softwares as well as very user friendly. This is what we can do. There are some disadvantages. The reproducibility will be not there in most of the times. This is a major disadvantage of this 2D electrophoresis. And again, softwares, they are very user friendly. Still, there is lots to need to be developed so that we can use very effectively.
So, as you rightly mentioned, so two-dimensional electrophoresis is still a very powerful tool to resolve thousands of proteins on the gel.
Yes.
One can also analyze isoforms and post-translational modifications, including phosphorylation, and overall the process is very user-friendly. Yes. So, still it is one of the very powerful technologies currently available for proteome analysis.
Yes.
Now, I also agree with your comment that there is some reproducibility issues because users have to run the gels from the control and treatment separately, and then there will be a lot of manual artifacts regardless of how meticulous they are doing the experiment. So in that regard, what is your suggestion, like how user can overcome the limitations of traditional two-dimensional electrophoresis technology?
One should have to prepare very good sample preparation so that the reproducibility thing will overcome. The second point is, these days the new generation 2D electrophore system, that is the DICE technology, is available, where you can use 3 samples in a single strip, and we can co-migrate them in a single strip, and we finally there will be an internal control as well as control and treated. These 3 you can run in a single strip, so that these kind of reproducibility issues we can overcome. As well as there is a scanner, very powerful scanner is available in this technology, that is the Typhoon Trio, where you can have this is based on the laser based technology, is completely, so that you will have very efficient gels you will have in your hand. This is
so as you mentioned that DICE technology is one of the very powerful solution to overcome several challenges which people face in running traditional 2D gels. And so in that regard, I think if you can elaborate a little bit about DICE technology, so that will be useful to the students.
Yes, I have a small video regarding the DICE technology. I'll show you that video so that everyone can able to easily understand what exactly DICE technology. Yes, as we were talking about the 3 samples we can label with Psi2, Psi3, Psi5. Now you have 2 different samples from different source. Now you are taking these 3 samples into 3 different Eppendorf tubes, and you are labeling with Psi3 dye as well as again another with Psi5. Then you are taking those 2 samples, pooling them and labeling with Psi2. Then you're mixing those 3 into a single tube, then you are running in a single strip. There you will be analyzed in first dimension the IEF, whereas in first dimension after analyzing IEF you will be placing into second dimension, where you are completely analyzed the second dimension. Now this gel can be scanned in the Typhoon Trio Plus, where you will have 3 images from a single gel. Then this can be analyzed with the help of decider software, then where the complete analysis can be done with the help of decider. Then in DIA there will be a co-detection. The co-detection nothing but there is a standard gel which is labeled with Psi2 from the same area we are expanding remaining 2 images also, so that the artifact can be minimized. Again, this is fully automated analysis. There will be very less manual interference would be there in this analysis. And after finishing of DIA, we can go to BVA. In BVA, we can compare all DIAs together. Like as you can see in this video, this complete first of all it matches between the standard gel, and from standard gel to again the corresponding DIA. It matches each and individual spot, and it will give you the final data. Then these BVAs can be analyzed in the further in EDA, that is extended data analysis. Here you will have lots of stringent statistical data, that's a differential expression analysis, pattern analysis, discriminant analysis, and PCA analysis.
So Dr. Srinivas, it was very useful to get the glimpse of the overall process in dye-etch technology. Would you like to elaborate or demonstrate some more detail about steps involved in doing the labeling of this technology?
Yes, there is another video where you can see now. This is the second video which you can see more elaboratively. This is particularly a protocol which is developed for membrane protein analysis. Now you can see this video.
In the cell surface labeling protocol, seen on top, you label the cells while they are still intact. During the labeling process, the dye will only have access to the cell surface proteins. After the labeling step, the cells are lysed. To verify cell surface specific labeling, the labeled sample was fractionated into membrane and cytosolic proteins. A non-fractionated sample was prepared in parallel for comparison. This fractionation analysis is not necessary, but was done here just to show that the cell surface protocol is specific for cell surface proteins. We also performed the standard etan dye-etch labeling protocol, seen below. The cells are lysed before labeling, and in this way all cellular proteins are accessible for labeling. After the labeling step, the samples are subjected to 2D electrophoresis. Adherent cells are detached using a non-enzymatic procedure to avoid digestion of the cell surface proteins targeted in this protocol. We used a rubber policeman, but using enzyme-free cell dissociation media is also an option. Count and divide the cell suspensions into aliquots of 5 to 10 million cells per tube. The cells are then pelleted and washed in HBSS pH 7.4 to remove traces of cell culture media. Contamination from serum proteins and fluorescent components can interfere with labeling and detection. Cells growing in suspension are directly pelleted and washed before the labeling step. After the wash, the cell pellet is resuspended in 200 microliters ice-cold labeling buffer containing HBSS pH 8.5 and 1 molar urea for optimal labeling conditions of cell surface proteins. Always check pH before labeling. We used 600 picomoles Sci-dye for 10 million CHO cells. The optimal ratio of Sci-dye to cell number will vary depending on the cell type, since we do not know the exact protein concentrations on the cell surface. How to determine the optimal conditions for Sci-dye labeling of proteins is described in the 2D electrophoresis Principles and Methods Handbook. The cells are incubated with Sci-dye dye-etch floor minimal dyes for 20 minutes on ice in the dark. After the labeling reaction, the unreacted dye is quenched by adding 20 microliters of 10 millimolar lysine. The labeled cells are now washed twice in cold HBSS pH 7.4 buffer to remove the excess Sci-dye. There will therefore be no free dye left for unwanted intracellular labeling of proteins in the next step, which is cell lysis. The proteins on the cell surface are now labeled and the cells are washed and ready to be lysed. The pellet from the last washing step is resuspended in 150 microliter cold lysis buffer containing 7 molar urea, 2 molar thiourea, 4% chaps, 30 millimolar tris, 5 millimolar magnesium acetate pH 8.5 and left on ice for at least 1 hour with occasional vortexing. The samples are now ready for 2D gel separation. The first step in 2D electrophoresis is to prepare IPG strips for rehydration. Prepare the streak rehydration solution by adding IPG buffer corresponding to the pH interval of the strips used, and add the solution in the lanes of the rehydration tray. Remove the protective film of the IPG strip and place the strips carefully with the dried gel facing down in the rehydration tray containing the rehydration solution. Close the lid of the IPG box and rehydrate the strips overnight. In the first dimension, isoelectric focusing, the proteins are separated according to their pI. This is performed using the IPG-4-3. The rehydrated strips is placed in the manifold and the electrode is mounted on top. 50 micrograms from each sample were applied using sample application caps. We have either directly applied non-fractionated samples without prior fractionation or fractionated the samples into membrane and cytosolic fractions before they were applied. The lid is closed to protect the fluorescent samples from light. The instrument was programmed according to recommendation and run overnight. Large 12% Lemley gels were cast using a Dolt 12 gel caster. Displacing solution was added to avoid polymerized acrylamide gels in the tubings. The gels were allowed to polymerize overnight at room temperature prior to use. After the isoelectric focusing, the strips are removed and equilibrated in SDS-containing buffer in 2 steps using DTT to reduce the disulfide bonds of cysteine residues, followed by alkylation with iodacetamide to avoid modification by acrylamide. The IPG strips are dipped in running buffer and carefully placed on top of the large 2D gels. Avoid trapping air between the strips and the gel. Seal by adding melted 2% agarose solution with bromophenol blue on top. The gels are now ready for second dimension SDS page separation. In the second dimension SDS page, the proteins will be separated according to molecular weight, and this is performed with the etan Dolt 6 system. Fill the electrophoresis chamber with anodic running buffer, insert the gels and fill the top compartment with cathodic running buffer. Program the power supply according to recommendations and run the second dimension protected from light for approximately 4 to 5 hours or until the dye front reaches the bottom of the gel. After the second dimension electrophoresis, The gel cassettes are placed by using the grippers in the Typhoon fluorescent imager. 2 gels and 3 channels can be scanned simultaneously. The result from this 2D gel shows high resolution of membrane proteins in the sample, even if there are some known restrictions for hydrophobic proteins to be detected in a 2D gel. The results show many new cell surface label spots, shown here in red, that are not detected using the standard labeling protocol, shown here in green. These results show that the cell surface labeling protocol is highly specific for labeling cell surface proteins. Since cell surface proteins are exclusively labeled, they are more easily visualized and attenuation by abundant cytosolic proteins is avoided. The fluorescent image of gels with non-fractionated, membrane fraction or cytosolic fraction is shown on top. Below is an image of the same gel post-stained with silver. The results show no fluorescent labeling of cytosolic proteins, but the silver staining shows that there are proteins in the gels. The results also show similar spot map patterns from non-fractionated and membrane fractions, demonstrating that there is no need for fractionation prior to 2D electrophoresis, which makes this protocol both simple and convenient. Cy2, Cy3 and Cy5 show similar labeling patterns and are all compatible with the cell surface protocol. A Deitch experiment was performed using all 3 Cy-dye Deitch fluor minimal dyes for studying differential expression of cell surface proteins in CHO cells after serum starvation for different lengths of time. Cy2 cell surface samples from all samples in the experiment were pooled and used as an internal standard. The differential changes of several cell surface proteins could be followed during the starvation period. Over 80 novel membrane proteins were detected using the cell surface protocol that were not detected with the standard Etan-Deitch protocol. For finding the identity of the proteins in the preparative gel, it was necessary to spike with a cell surface sample to facilitate matching back to the spots on the analytical dataset.
So Dr. Srinivas, it was very useful to see all the steps which are involved in doing this Deitch technology. There is that discussion going on in the proteomics field that due to advancement in mass spectrometry and microarrays, how do you foresee the future of two-dimensional electrophoresis and Deitch technology? Do you think it's going to be simultaneously working along with the mass spec and microarrays, or do you think it's going to be behind because now more advanced approaches have emerged? Yes. So what is your suggestion on that?
Yes, the MS and microarrays develop differently. They have their own advantages as well as disadvantages. Like disadvantages like very high instrumentation cost and high protein requirement, as well as technical variance would be there. That is why still 2D Deitch will be the core technology to analyze proteomics. One can easily analyze.
So you are still very convinced that by using 2D Deitch approach, that will still remain one of the core proteomic technology for proteomic analysis?
Yes, definitely.
And it will be used simultaneously along with mass spectrometry and microarrays?
Yes.
So at the end, I would like to request you to give a brief overview of the Decided software which is being used for analysis of Deitch gel.
Sure.
Decider is the software to analyze Deitch gels, where we can analyze Deitch gels in different stages. In first stage, we have to upload our gels into our software. This is basically works on the Oracle database, where we are incorporating our gels into database. There you can add these gels into software by this way, and you can select what are the gels you want to upload, and once you have uploaded, then you can able to edit these gels here itself, like you want to any crop or edit anything, any sort of things you can do here. It will allow you to edit these images. So now you can see this is the overlay of 3 images. Now you can crop all these images, like whatever the portion you want to take. If you have to remove any area which is not your interest of, then you can directly crop those images. By this way, once you directly editing, you can save it, yes, save it and close it. This is what you can edit these images. Then you have to create a new project. Suppose your project name would be according to your project name. This is demo. Okay, now we have the project name of demo. Now you can able to import these 2 gel images into a new project where we created. Just click on import. Now these gels will be imported into this particular project. Now here you can see all 6 images. Though these are the basically 2 gels, inside within these 2 gels we have Sci2, Sci3, Sci5 and total 6 gel images. Okay, this is the image editor and image loader. The next one is differential in-gel analysis, shortly we call as DIA. In this you can see we can create a new project, new DIA here. Like there is an option create workspace. From here you can, now it will take you to where we already saved our gels in our database. Now from the database we can select any particular project, and from there I am selecting gel number 1 as we saved this one. Then now you can see this gels where we have uploaded. So now after uploading here you can process these gels. Then during process you have to give some number. This is some threshold which you are giving here. Actually this may be 2000, so that it will takes care of background issues also. Then basically in decider code detection will be happens. But I would like to explain you some more about what is code detection. This code detection is uses a information of all 3 channels and will create a geometrically identical spot boundary for a spot across all the channels. That means there are 3 channels, Sci2, Sci3, Sci5. Out of these 3 channels, in Sci2 image it creates a particular volume, and the same area can be applicable for the remaining 2 gels also. This is way it works. In this way, quantitative and qualitative results are much more accurate than with a single detection. In DIA, each image is co-detected with its internal control, producing 2 images pairs. The ratio of standard sample is calculated further, or the ratio of standard sample is calculated for each protein in each image. So as we see here, these are all the number of spots which it has been detected, in which there are, you can see some red color spots here. These are all down regulated and compared with control with treated, or and these blue colored spots where you can see these are all upregulated spots when comparing with control and treated. In between this there is a blue, there are some blue colored spots. These are all similarly regulated. So this is what we can see in DIA. Now you can go through each and individual spot and you can see the 3D view of that particular spot. If suppose if you can select any one, you can go to one by one and you can see whether it is exactly spot or some background, or if it is a background you have to remove that. So if suppose this is the background, so there is no spot at all, still it is detected some background, so you can exclude it from here by clicking, then confirm it. So, this protein has been removed from the gels. So, the same way you can go each and individual, then exclude it, confirm it. By this way you can check all spots and you can have more accurate data with you. Like you can see the how accuracy would be there, like the spot detection. Now you can see this is what we will get in DIA. This DIA creation we finished. This is the BVA. BVA is nothing but biological variation analysis. One of the internal standard image is selected as a master image and all internal standard image matched into this. Samples standard spot ratio for each protein, each sample, then compared giving t-test value, fold changes, ANOVA values for each and individual protein. How to create BVA workspace? Can open the BVA and create a BVA workspace and go to our DIA workspace where we have our DIAs. From there you can create, actually add it. One DIA I have added and this is another DIA I am adding here. Minimum 2 DIAs we require for the BVA. So, we have 2 DIAs here and click on create. So, it creates a new BVA for you. Okay, this is the new BVA. From here the first all Site2 gels are automatically goes to standard folder. There is a standard folder you can see, and remaining all gels remains in the unassigned folder, where we need to assign these gels as according to the gel type or sample type. Then you just have to click on add option. Now create a group, it may be control or treated. This first one is the control, and apply some color draft. Confirm it. Then another you can create like treated, then give some color draft. Confirm it. Now we have two folders, control and treated. So, as we have in assigned folder both control and treated, this control gels we can transfer into control folder by dragging those images, and treated gels we can transfer to treated folder by dragging them. Now we have our images here. After shifting the control to control and treated to treated, we have to match all gels. Just click on match and match all. Just match. The matching process has been finished. Now, as we discussed, in out of all standard gels, one gel selected as a master image. As you can see, the number 24 gel has been selected as a master, and it will compares remaining other gels with this master gel. So, now we have this comparison data. After that, we need to calculate statistical parameters. So, click on statistical parameter button. See, now we have some statistical parameters like independent t-test, average ratio, student t-test, one-way ANOVA in between different groups. We are doing in between control and treated. So, calculate them. So, the statistical parameters has been calculated. Now, we can see exact results of statistical parameters. If I can go to the table view, here you can see t-test, one-way ANOVA, this you can see. So, we can select from here which are all the statistical significant and which are all not significant. After analyzing the statistical data, now we can see the complete results here. Here we can see the 4 views, like this is the image view and this is the histogram view. In histogram view, we can see clearly particular protein how it is behaving throughout control and treated. If you can see, this is the standard gel, that means this is the mixture of these control and treated. So, it is somewhere 0 we can consider this one. Then the control is completely showing upregulation, and after giving the particular treatment, it is showing the down regulation. So, this kind of data we can see here. Then in the table view, as you can see the complete protein data, where the t test value, average ratio value, 1D ANOVA value, this all we can see we can see here in the table view. So, the 4 views at a time to see, this is the 4 views we can see here. So, after this we can filter them according to our interest. So, select the few parameters which are available, restrict to 10 gels which are spot maps which are present, a particular protein should be there, and student t test as well as average ratio, then one way ANOVA value, then filter it. So, there are 2299 spots are available in this all gels, but 107 proteins only passed all these parameters. So, these parameters we can select as a protein of interest and assign pick list, so that these proteins can be saved in a file. This file can be given furtherly to our spot picker. These are all the things we can able to identify very easily in BVA. This is very user friendly. There is no much more manual interference. Okay, this is what this helps you to analyze your dye gels.
So, can you elaborate on what is EDA or extended data analysis, what it can do, which we are unable to do it in BVA? So, there is a layers here, right? One is the DIA followed by BVA, and then ultimately EDA analysis.
Exactly. Basically, what we can do here is we can compare two BVAs together there. Exactly. Here we are talking about a particular disease or particular set of data only. There we can analyze different BVAs together in DDA. There you will get majorly differential expression, again you will get, as well as PCA and discriminant analysis. You will, these kind of statistical data you will get in EDA. Very shortly, just show you briefly regarding.
So if I understood correctly, probably the statistical parameter will be more stringent over there in the EDA, and you can have some better biological significance information from the data set.
Exactly.
Because in a lot of clinical data or different type of treatments, people like to do several gels and lot of treatment.
Yes.
So your number of sample to be analyzed is very large.
Exactly.
And really obtaining the meaningful information is one of the major challenges in all the proteomics field. So I'd like to see in an EDA.
Please. Just. Thing which we can see here is differential expression analysis, in which you can see differences in between control as well as two experimental data. This is the different treatments which given. Here you can see how the particular protein is expressing throughout these control as well as this PF and PV, these kind of things, and you can see this kind of data for each and individual protein here, so that from here you can see which one is your interest and which is not
so
your interest.
You are actually analyzing the data spot wise now, spot by spot. You are looking at
exactly. Here the spot by spot which we are seeing, the number of the the index number which shows there is a master gel. From that master gel you can see that exactly this number. This is what which we are seeing here for each individual spot. Here we can see the results. Then as well as if we can see principal component analysis for this data. here there are almost 89 proteins. Out of these 89 proteins, you can see there are inner the circle there are proteins, some proteins are present, and outer the outliers are there. The inner circle they are similarly, especially if I can say 95% statistically significant there, and outliers which you can see are exactly this can be some non-reproducible spots, or else what the major thing is these are very highly upregulated or highly downregulated. So this can be worked as a marker also. Then we have to go back to our BVA data and we can check that protein how exactly it is regulating. Then we can identify that protein and we can use for further analysis.
So this is a powerful statistical parameter
Exactly.
by using which you can identify some outliers, which
Exactly.
could be the potential discriminator between the
Exactly.
control and treatment. And once you identify those proteins, then actually you can go back to your original data from the BVA and get all the analysis done.
Exactly.
Oh, this is very
can be done.
interesting.
And the next pattern analysis, you can see the whole proteome, then how these are different from each other.
So this is the heat
See,
map of
this is
data.
the heat map of the total 82 proteins are which we take into consideration. Then how in which area they are upregulated. If you can see the blue area, the green area exactly we can see is completely downregulated area, and the red color portion where you can see that is the upregulated portion, and the remaining black color which you can see. So those proteins are similarly regulated. This is what which you can see here. This kind of data will help you to represent your complete whole analysis.
So Dr. Srinivas, it was very useful to have you here and to get an overview of DICE technology, how people can use decided software and analyze their data by using DIA, BVA and EDA. And although there was not enough time, but you gave a very good demonstration in a very short time to give a glimpse of the processes involved for doing this analysis, as well as how different type of statistical parameters can be applied to get some very powerful statistical information from our biological data. So thank you very much for coming here and giving this very good introduction about DICE technology. So I hope our discussion with Dr. Srinivas was useful, and now you can perform these analysis by using a specialized software and obtain some very useful biological information from your data set. Probably you must appreciate there are a lot of meticulous steps are involved in performing these experiments, but at the end this provides a very useful quantitative multiplexing approach to separate proteins and to analyze different type of variations. So, in summary, in this module we have discussed about gel based techniques. We talked about basic separation by using SDS page, blue native page, then how to perform isoelectric focusing, and by combining those two methods, how to perform two-dimensional electrophoresis. I gave you a workflow for performing two-dimensional electrophoresis experiment. Then we have discussed about different type of challenges for these methods, and I briefly introduced few creative ways of performing gel based proteomics method, including off gel fractionation methods, and then we discussed in much more detail about difference gel electrophoresis. I hope at the end of this module and lecture you will be able to perform the gel based proteomics experiment. But please keep in mind, these protocols and methods are only giving you a feel for performing these experiments. Each experiment, each sample type, each biological question brings its own unique challenges, and depending upon those conditions and your sample type, you need to optimize these methods. There is no one technology which can answer all of your question, but it is good idea for you to know that what are different methods which are available for you to use. So, I hope by taking this module on gel based proteomics, now you are familiar with different type of gel based techniques These are only few, there are many other methods as well available, but these are the most commonly used methods which people are applying in the fields of proteomics. Thank you.